Hi, I would like to present our study on drug discovery for serine-glycine 1-carbon pathway enzyme SHMT2 by deep learning and transfer learning. Serine hydroxymethyl transferase 2 SHMT2 in the serine-glycine 1-carbon pathway is a key enzyme with significant therapeutic potential for idiopathic pulmonary fibrosis (IPF) and other fibrotic diseases. SHMT2 is required for the production of collagen, and its overproduction leads to loss of lung function in IPF. Our objective in this study is to find drug candidate compounds for SHMT2 by virtual screening that uses deep neural networks and transfer learning. There is not any study on drug-target interaction prediction and virtual screening for SHMT2 due to the limited number of active compounds against this enzyme. Deep learning-based virtual screening methods are very successful for virtual screening. However, they require a large number of training data. To overcome this problem, we explored transfer learning along with deep learning. We use two modes of transfer learning. In the first mode, we use the pre-trained source model as is, which is also called full fine-tuning. The whole trained source deep neural network is further retrained for a few number of epochs, as it is fine-tuned with target task dataset, which is of limited size. The source network provides an initial configuration for the target network. Therefore, we don't start from random configuration. In mode 2, we do fine-tuning with freezing layers. In fact, the bottom layers of the source network are frozen, and these frozen layers correspond to the layers where low-level features are extracted. During training of the target network, the weights of these layers are not updated. In fact, what we do here is to retrain only the final classifier layers. Here is the algorithm that we follow. First, we extract the compound bioactivity data of SHMT2 from Campbell database. Then we construct prediction models which are composed of feed-forward neural networks using transfer learning. The source model here is a feed-forward deep neural network model trained for proteins from an upper level class family. And the target models, we obtain the target models by retraining the source model for SHMT2 with the two modes that we have mentioned earlier. And finally, we found compounds similar to active compounds of the target protein and screen them via trained feed-forward neural network models. The deep neural network model that we use is a ligand-based binary classifier, which is composed of two hidden layers, in addition to the input layer of 1024 neurons and an output layer of 1 neuron. In fact, 1024 neurons correspond to the fingerprint of the enzyme. After hyperparameter selection, the number of neurons in the first hidden layer is 496, and second hidden layer is 1024 again. The source data we obtain from the Campbell is an enzyme with EC number 2.1.2.1. So we selected enzyme classification with number 2.1 family to form the source data set. And with Campbell, we obtained 732 active and approximately 1900 inactive bioactivity data. We apply 5-fold cross-validation to generate the source model. The target data is also extracted from Campbell with the target enzyme SHMT2, and there are 6 active and 179 inactive bioactivity data. The performance of the the target model is measured with the leave one out method, since the number of bioactive data is very low. We assess the performance by using the matrix correlation coefficient for the models. The source network model MCC is 0.713, and the target network models of mode 1 is 0.267, and for mode 2 is MCC is 0.325. Once we have obtained the ligand-based binary classifiers, we screen the compounds from the drug bank against SHMT2 by using the trained target models, and we took the consensus of the two models, and we found 4 approved drugs and 8 experimental drugs to be candidates against SHMT2. Now, in fact, what we have done is to use transfer learning to find drug candidates against a protein having very few bioactivity data, and we will consider further other proteins in the serine-glycine 1-carbon pathway. Thank you for listening.